Welcome to the first lecture in this series of lectures on the medical benefit of cannabis-based medicines. In this first lecture, I'm going to talk to you about clinical trial design and critical analysis. So you might ask yourself, why do I need to know this? Why is this important in the understanding of the medical benefits of cannabis? Well, there are several reasons. The first is because I'm going to talk to you a lot about the clinical trial evidence that we have that cannabis-based medicines are useful in a variety of different disorders. And in order to do so, I'm going to discuss various clinical trials with you. And it's important that you understand the terminology, and so you can understand the clinical trial design and what that evidence tells us. It's also very important to be able to understand the types of trials being covered throughout this model and what the data tells us about the effectiveness of cannabis-based medicines. And to do so, you have to understand clinical trial terminology. And it's important that you start to critically analyse clinical trial data. So not everything that's published is the best that it could be. And with hindsight, we can see that there are strengths and flaws in every piece of work that's been done. So it's important that you can begin to be able to look at a piece of evidence and make some judgments for yourself about how good you think that evidence is, what is the strength and the quality of the data, and therefore make a decision about what you think the overall strength of evidence is in a particular disease indication. So that's what I'm hoping that you'll learn from this lecture. So first of all, let's just define what a clinical trial is. A clinical trial is any research study that uses human volunteers, and that can be people who are healthy, but most commonly it's patients. And it is designed to determine the safety and effectiveness of something. Now, most of the studies that we'll be covering are determining the safety and effectiveness of a drug or a medicine, which is cannabis-based medicines or anything that is a cannabinoid-based medicine. But clinical trials also cover other things like vaccines, biologics, devices, that could be prosthesis, but it could also be an inhaler or a vaping device, and things like behavioural interventions. So can a change in diet or exercise affect an endpoint? So effectively anything that involves a human which is determining the clinical safety and effectiveness of a new medicine or behaviour or device is called a clinical trial. And this is how we determine whether or not we think something works. There are several phases of clinical trials. So, well, first of all, let's cover the types of clinical trials. So the most common that we're going to come across is called an interventional or treatment trial. And this is where a new experimental treatment, such as a cannabis-based medicine, or it could be a new combination of drugs, it could be adding in a medicine on top of standard care, or it could be new approaches to surgery or radiation therapy. Anything like that, which is where you are intervening with a new treatment, is called an interventional trial. You can also have prevention trials, and these look for ways to prevent diseases from occurring, and it usually involves things like new medicines or vitamins or vaccines or minerals or lifestyle changes. So can we stop a disease from developing in the first instance? Then there are diagnostic trials, and these are studies that are trying to find better tests or procedures for diagnosing a condition. Usually, the quicker that you can diagnose a medical condition, then the better your outcome will be and the better your treatment options will be. And there are also screening trials. So these are clinical trials that are looking for the best way to detect certain diseases. So you can have cancer screening trials. We know lots of people screen for cervical and colon cancer, or lots of countries screen for these types of cancers to try and detect them early, again, because your outcome is going to be a lot better the earlier that you detect a disease. And then there are also quality of life trials. So these are also relevant to cannabis-based medicines because they're exploring ways to improve comfort and the quality of life in chronic illnesses. So you're not necessarily looking for a cure or a treatment, but you're just trying to make people more comfortable with their lifelong condition. So there are several stages that you have to go through in order to develop a clinical trial and to really get to the definitive answer of whether or not you think your treatment, because I'll talk mostly about interventional trials, is going to be effective or not. Before you can even begin to think about doing something in humans, you have to have what's called a preclinical phase. And this is where you do laboratory experiments based in cells or in animal models, where you determine whether or not your medicine is going to be safe and effective in a model of that disease. And only when you have data that proves that in that preclinical phase, will you get ethical approval to then move that into humans. So you have to have a very sound reason for thinking that your medicine is going to work. So you have this preclinical phase of development which comes before you even go into humans. Then you have your phase 1 trial, and this is the initial introduction of a new drug, and it's usually carried out in healthy volunteers. So this is a compound or a chemical that's never been in man before. So these trials are quite risky because you don't know what kind of reaction the human is going to have. You've given it to animals, but you don't know whether or not the response will be different in a human. And that's why we normally do this in healthy volunteers rather than in the patient population. With cannabis-based medicines, we don't tend to need phase 1 trials if we're using the chemicals from the plant, because we know that these chemicals have already been in humans before, and so we're not so concerned about the potential safety. Now that might be different when we start thinking about minor phytocannabinoids where humans haven't been exposed to very high levels of these compounds. So once you've determined that it is safe to give your new drug to a healthy volunteer, the second phase of a clinical trial is to evaluate the effectiveness and safety in relevant patient populations. So really the key word here again is safety, but also now, does it work if I give my medicine to relevant patients? So if you're trying to develop a new asthma drug, then clearly in phase 2, you're going to now give your medicine to patients who have asthma to test, does it work against whatever outcome I'm trying to measure? And is it going to be safe in this patient population, which is the population I want my medicine to be developed in? When you get a positive outcome in that phase 2 trial, so you get a signal that your medicine is working, then you go into much bigger, large controlled studies. And you only do this when you get a signal from your phase 2 that the medicine is safe and is working. Then you start looking at it in much bigger populations, usually in a multi-center trial, so using not just one hospital, but lots of hospitals, preferentially across the world, so that the outcome that you get is global. It's an international outcome that no matter where, whatever the genetic background is of your patient population, you still see the same type of response. So phase 3 are the really critical trials, because these are the ones, if you get a positive result in a phase 3 clinical trial, that's where you can then go to regulatory bodies and ask for your drug to be licensed for whatever it is that you've proven it works for. So sometimes you can get this after one large phase 3 trial, but it's more common that you need to do this a few times. You need to prove it a couple of times before a regulatory body would say, okay, we think your medicine really does work, and so we will license it to be prescribed. After that licensing phase, you can have an additional phase. Not all medicines go through this phase, but you can have what's called phase 4 clinical trials. And this is where we gather a lot more information about that medicine being used long-term in a patient population. So most phase 2 and phase 3 clinical trials are quite short in duration. They might only be 2, 3, 4, maybe 6 months of treatment. But now you're going into populations and you're potentially prescribing your drug for years or for the rest of their life. So you need to know, are there additional risks that we didn't see when we were doing these kind of shorter clinical trials? are there additional benefits that we didn't realize? Are there other endpoints that are really improved by this medicine that didn't come out in the phase 3 trial? So we learn a lot more about the safety and benefits of a drug when it's being prescribed in very large populations for longer term. So sometimes that means that drugs can be withdrawn from the market. And I'll be telling you during the course of this module about a medicine called rimonabant, which was developed to try and block the CB1 receptors in our brain as an anti-obesity drug. And it was very effective, but once it got to past licensing and was being prescribed in a broader population, what they found was that blocking CB1 receptors in our brain actually has quite a negative impact. And there was incidences of depression and suicide. So it wasn't long after licensing that actually this medicine was withdrawn from the market. So that often happens when you go into a broad population. So they're the phases of clinical trials. And let's just look at this in a slightly different way. So again, just focusing on the phase 1, phase 2, and phase 3. So what is the question? In a phase 1, the question is this treatment safe? Can I give this chemical to a human? In phase 2, we're asking, does it work if I give it to the right patient population? And in phase 3, we're asking, does it work if I give it to large numbers of the right patient population? The risk level of phase 1 studies is really quite high. This is the first time you're going to be exposing a human to this compound, and there can be negative impacts. And again, in cannabinoid-based medicines, we have quite a famous example here of a trial a couple of years ago, a phase 1 trial, where the investigators were giving a FAAH inhibitor. And the FAAH is the enzyme that normally breaks down the endocannabinoids in our body. You might have learned about it already. And they had developed an inhibitor of this enzyme, which increases our endocannabinoid tone. But when they gave it in their phase 1 trial, it actually had really significant side effects in the patients. And one of the 6 subjects unfortunately died, and the other 5 ended up in intensive care. Now, it turned out it was because this drug did more than they had realized, and it had what we call off-target effects. So it did more than just inhibiting the FAAH enzyme. But that just gives you an example of the fact that these trials are really risky. So this was, you tend to only give the new drug into a very few participants when you're doing your phase 1 trials. Phase 2 trials are also risky because it's the first time you're going into your patient population, but we do know a little bit more about the drug at this stage, so it's a moderate risk. Similarly, in phase 3, the risk is slightly lower because we've already exposed quite a few humans to your medicine, and we know a lot more about the safety of the drug and how well it works. But this is where sometimes if there is a side effect which has a low incidence, so if you have a side effect that only maybe comes out, has a 1 in 50,000 chance of occurring, you're only going to see that side effect when you start giving your medicine to lots of people. So there is still a risk in a phase 3 trial. In terms of the length of trials, phase 1 trials are usually quite short. So we start with a single exposure to a drug. If that goes well, then you might do multiple doses over maybe a week. So very short exposure. Remember, it's quite risky. Phase 2 and phase 3 are usually between 1 and 3 months. That's the typical length of a clinical trial, because in these trials, you're looking for an effectiveness of a medicine. So you're looking to see a change in an endpoint, which normally takes a certain amount of time to develop. In terms of the number of participants involved, there's usually only a couple of people involved in phase 1 clinical trials because they're so risky. When you get to phase 2, it's maybe up to 100 participants, depending sometimes on how many different groups there are in the clinical trial design, and we'll come to that. And in phase 3, you're normally talking about much bigger patient populations, so hundreds of patients in these kind of studies, because that's where you're getting your more definitive answer of, does the drug really work in large numbers? Now, we have quite a lot of clinical trial data with cannabis-based medicines that sits at phase 2. So there are a lot of kind of small, we call them proof of concept trials, where the effectiveness of a cannabis-based medicine has been tested in small patient populations, maybe up to 50 patients, but we would still consider that to be, a limited amount of evidence about the effectiveness of the drug. It's really only when you see positive outcomes in the phase 3 trials that you really develop certainty that a medicine is really working. Now, the problem we don't have so many phase 3 trials with cannabis-based medicines is that they're very expensive to run. So phase 2, it's smaller, so more manageable, but phase 3, they tend to be international, multi-center with hundreds of patients and cost an awful lot of money and tend to only be funded by companies who are trying to develop a particular product as a licensed medicine. So we have a lot fewer phase 3 clinical trials with cannabis-based medicines for that reason. So this is a typical drug discovery pipeline. What I'm describing to you is the pipeline of how a medicine goes from that initial kind of concept of being effective in the laboratory and in animal models to progressing into humans through various phases of trials and then being approved at the end, which is what you really hope will happen with your medicine. A couple of things to point out from this figure is that the typical drug discovery pipeline takes quite a long time, so 12 to 15 years from initially having that kind of laboratory idea that something is going to work to getting an approval. Now, in cannabis-based medicines, that can be a little bit shorter. So if we consider the story of Epidiolex, the purified CBD medicine, which is now licensed for patients that have rare forms of epilepsy, that entire drug discovery pipeline was actually a lot shorter because it started really with phase 2 trials. So a lot of the initial phases could be skipped, and it went quite quickly into phase 3, and they did 4 phase 3 trials in a short amount of time, and then they were able to apply for approval. So I think the entire process probably only took about 6 to 8 years. So it is possible in cannabis-based medicines with a novel product to be able to actually get through that pipeline quicker than you would in a normal drug discovery process. The other thing to point out is just how much money this costs to do. Particularly in terms of safety package, so being able to prove that a medicine or a drug is safe in animals and then in humans really does cost quite an awful lot of money. And similarly, once you go into the phase 3 trials and doing multiple phase 3 trials, you're talking about nearly billions of pounds to develop a medicine.